So we have the 2020 HSC Biology paper, question 1 to 20. So for this, our first question is looking at homeostasis. And essentially, it's looking at a behavioral adaptation. So what we're concerned with is going to be something that you can do voluntarily, and it's also what you do naturally. So in this case, option B will be correct, because this will help you stay warm. So it's an example of homeostasis for temperature. This is correct example. However, it doesn't include a behavioral adaptation. That'll be physiological. Goes for the same with this one and this one. Question 2, we're looking at asexual reproduction used in plants. So this is going to be option D. So option A is a bit tricky, but the trick here is dispersal of seeds. Pollination occurs before the dispersal of seeds, because you need to have things pollinated in order to form those seeds. Cloning is going to be an example of asexual, and mitosis is going to be an example of asexual reproduction as well, because you'll have identical genetic information. So this is just an order question where you really just need to memorize this sequence. So our answer is going to be option C, and that's because we've got ovulation, and then for that to then occur, the egg needs to be fertilized, which will allow it to then be implanted. And after the cell has started to divide and divide and divide, we'll get some placental formation occurring. Question 4, we're looking at an infectious disease. So malaria is a example of a plasmodium species, which is caused by a protist, which is a type of parasite for humans. So for question 4, the answer is going to be C. So this is true. A mosquito is a vector because it's just carrying the plasmodium, and it will inject it into the human from its salivary duct. So when it actually bites humans. And as a side note, this is only done by female mosquitoes. And it's also true that the plasmodium is the pathogen because it's the thing causing the disease. So for question 5, we're looking at the difference between prokaryotic and eukaryotic DNA. So prokaryotic, we've seen before that it's circular. So it's either A or B. And then we know eukaryotic, it lines up in chromosomes. So therefore, it's got to be linear. So option B is correct. So this one's looking at a particular type of bacterial disease called citrus canker. So you're not actually expected to know this one. You're looking at your ability to work it out. So the option that's correct is going to be C. And that's because this will prevent the disease from coming into Australia in the first place. There's no use in treating it if you can prevent it. So for question 7, we've got an experiment. So we're looking at working out which condition is true. So the thing we're changing, which is our independent variable, will be the food sample. This tells me A or C could be correct. The dependent variable, or the thing we're measuring, is going to be the number of microbes. So can't differentiate them. The experimental control is going to be option A. So an agar that isn't inoculated or doesn't have the sample. This option here is an example of an controlled variable. So for question 8, we're looking at quarantining for a non-infectious disease. So that's the trick there. The answer is going to be option C. And that's because a non-infectious disease, by definition, can't be passed on to someone from someone else who's infected. And it can only go from genetic causes or lifestyle. So for question 9, got a epidemiology study. Is going to be looking at the effectiveness of public education for slip, slop, slap, seek, and slide. Okay, option C is going to be correct. And the reason for that is before and after. All the other options aren't looking at whether or not the campaign has had any effect over a time course. So the other options are also plagued with poor sampling technique. So this is probably the best one out of the 4 answers. So for question 10, we're looking at a farmer who's using artificial insemination for his cows. So the introduction of a rare recessive allele means we're going to have it pop up in our population, and that's coming from another source. So that means this is going to be gene flow, because he's taken it from one pool and put it into another. For this one, retina and cornea both correctly are both a part of this. So the cornea is going to be option I and the retina option II. However, the cornea is refracting light. So option A is correct. And you can actually have cornea transplants. So the purpose of cloning in agriculture, well, it's not going to increase the frequency of recessive alleles. It's not going to preserve favorable traits in offspring. Oh, yes, it will. So it's not going to preserve. So for question 12, option B is correct. And the reason for that is it's going to be just trying to keep those same favorable traits in future generations. This isn't going to preserve any genetic variability, and it's going to not increase the combination of alleles in a population. Okay, so question 13, can't do. Question 14, we're looking at 2 alleles. Okay. So this is going to be an example of a incomplete dominance. And the reason for that is because we're looking at number of cholesterol, the levels. We're not looking at different types of cholesterol receptors. So these would come in here if there was one type that was a good one and another that was defective. However, that's not the case. And the fact they haven't told us the gender of the individual tells us it's also not going to be sex-linked. For question 15, bit of a bigger question, but basically we're looking at the effect of a new antiviral drug. So if we have a look down here, the correct answer is going to be option C. So essentially the triangles tell us the toxicity and the black tells us the inhibition. And we're using just one micromole per liter dose. So only looking here. So essentially you want the lowest toxicity for the highest effectiveness. So, yep, matches here, here, and based on toxicity alone, we can cancel A and D out. And then we're just looking at which one gives a better response. So this server 60% here, and that's just under 30% here. So that means option C is correct. So for question 16, we've got the difference between adenine and guanine. So remember the acronym at Gold Coast? So A and T, G and C. So these base pair together in DNA. So what we've then got is going to be option B, because essentially what they're saying is we've got adenine and guanine, and they're going to be totaling 50%. And that's because DNA is double-stranded. It is also true that it's got a helical structure, but that doesn't explain our data. Adenine doesn't pair with guanine, and yeah, nitrogenous bases. That's just referring to the base pairs, so the individual bases, so A, T, G, and C. That's quite broad. So for question 17, we're looking at Snips. So as you can see, 3 sequences and they're all the same. So we're after an explanation for why we don't see this. And whenever you see this sort of question, we're going to be looking at the idea that the SNPs are part of DNA that is not expressed. So these are going to be found as base mutations in at least 1% of the population. Therefore, we'd expect to find them in non-coding regions, because if these were in coding regions, they'd likely have an effect on function. If they weren't silent mutations, in which case we'd see a lot more mutations which were unfavorable, so they probably wouldn't really exist. So we can't do question 18, but for question 19, okay, so this will get a lot of people, but it's 6 chromosomes, and this means we got 3 homologous pairs. So the only one with 3 homologous pairs is option A. And then we're looking at meiosis. So this is going to form haploid cells. So it's got half the genetic information. Okay. And finally, question 20. Okay. So we're looking at the influence of genes and the environment. So for identical twins and biological siblings, these are going to be more affected by the genetic information, whereas the adoptive sibling will be a good indication of how the environment that the children have been brought up in is influencing the incidence of the disease. Okay. So now we're just going to read off our graph. So as you can see, the family environment in trait A is going to have a high impact, and the genes is pretty low. And that's because they're all pretty high and there's not that much of a difference here, proportional to the size. So then for trait B, there's a huge genetic impact here and not that much of an influence from the environment. It is going to be high and this will be low. So right now I've got option A and B both open. So we have to work out then on the next one. Okay, so this is going to be, both these traits are low, so therefore it's option A. Hey, I hope that's all right. Thank you.